And awesome. All right. So hello, as stated, my name is Brianna Ramirez, and I work with the Dr. McCall Lab in the School of Medicine and Education building. My project focuses on examining the effects of agricultural dust exposure on intestinal permeability and epithelial cell differentiation in mice. All right, to get started, I want to state a few facts about air pollution. So the World Health Organization, or WHO, states that there's about 7 million deaths per year due to air pollution exposure. This could be due to the development of chronic obstructive pulmonary disease, or COPD, and cardiovascular diseases. There are many different types of air pollution. Our lab is specifically interested in agricultural pollution that comes from concentrated animal feeding operations. These CAFOs can include dairy farms with cows, chicken farms, or what we're interested in, swine farms. So we're interested in swine farms because not only is pork in high demand, as stated by USDA, but the swine farm dust is known to be more abundant in ultrafine particles, endotoxins, and bacteria that could be detrimental to your lungs if exposed. With that in mind, these swine farm workers that are being exposed to this dust have to work extended hours to upkeep these farms. This constant exposure can lead to the formation of chronic lung disorders within these farm workers. But interestingly, these farm workers are also experiencing gastrointestinal symptoms alongside these lung disorders, such as nausea, diarrhea, and vomiting. However, unfortunately, many GI issues are underestimated in clinical studies. A good amount of these farm workers are migrant workers, and they may not be able to receive the help that they need. So symptoms usually go unreported until they are severe. That is why it's very important to try to identify the mechanistic properties of the interactions between both mucosal sites, the lung and the gut. This brings me to a term called the gut-lung axis, which is defined as the interactions between the microbiota and immune system of the lungs and the microbiota and immune system of the gut. This brings me to the overall aim of this project that will eventually lead to the progression of my own specific questions. So the overall aim is to identify mechanisms by which hog dust extract, which is the term I'll be using instead of swine, can disrupt intestinal homeostasis. We hypothesize that exposure to this HD will alter intestinal homeostasis and lead to gut inflammation. To start off with this broad aim, Dr. Melissa Crawford, in collaboration with Dr. Tara Nordgen, intranasally exposed male and female C57BL6 mice with either PBS or hog dust extract or HDE. First, they wanted to see whether the HDE exposure was causing chronic airway inflammation within these mice. To do that, they took the bronchoalveolar lavage fluid from the lungs of these mice and measured the total cell count and two different types of immune cells. What they found was that there was a significant increase in the total cell count here and also an increase in the neutrophil count within these mice. These two combined are indicative of chronic airway inflammation. This is great for us, not for the mice, but it's great because then we can now start to explore this gut-lung axis because we first need to make sure there's an effect in the lungs to this dust exposure before we can go into the gut. With the gut in mind, I want to give a brief intro into a specific portion of the gut that I am interested in, which is the intestinal epithelial barrier. So to get started, you first have your intestinal epithelial cells, which are your physical barrier in your gut. Within your physical barrier, you have tight junctions. These tight junctions regulate the pathway molecules from your intestinal lumen, where your poop passes through, into your cells. If these tight junctions were not to work or be affected, then you would get what is known as a leaky gut, meaning molecules that shouldn't be able to pass through are passing through, and that can affect the function of your barrier. Alongside that, we also have your mucosal or chemical barrier. Specifically, I'm interested in goblet cells as they secrete mucus that contribute to this barrier, and I will talk more about goblet cells later on. So going back, so now that the lab saw an effect in the lungs, they wanted to see if there was an effect in the gut in terms of intestinal permeability. So we have two probes, FD4 and RD70, that were gavaged into the stomachs of these mice. And we wanted to see if the presence of these two probes were in the serum of the mice. So FD4 is a much smaller molecule. If this was present in the serum, then that is indicative of a leak pathway or a tight junction dependent pathway, meaning your tight junctions are being affected. If RD70, a much larger molecule, shouldn't necessarily be getting through to your serum, is present, then that's indicative of an unrestricted pathway or tight junction independent pathway. So I bring your attention to the graphs here. We see not only a significant increase in the smaller molecule, smaller probe, but also in the larger probe. And these two combined can be indicative not only of increased intestinal permeability, but possible cell death happening within the barrier. Now that we saw that there was an increase in intestinal permeability, we also wanted to look at lipopolysaccharides or LPS, as they are in a greater abundance within this hog dust extract, and lipopolysaccharides are what give gram-negative bacteria their pathogenic characteristics, so it's very important to look at. And so here they wanted to measure the serum, the endotoxin levels or LPS levels within the serum, and there was a significant increase within these HDE treated mice, which is indicative of endotoxemia. Endotoxemia means you have higher concentrations of LPS that could potentially cause inflammation throughout your body. And so we may think that this LPS could be contributing to this increase in intestinal permeability that we were seeing. This then leads me to my questions of my project, which is one, is LPS contributing to an increase in permeability following HDE exposure? And two, does this HDE exposure also affect goblet cell differentiation? So are we seeing more or less goblet cells in the gut due to the exposure? To see whether LPS affects intestinal permeability, we looked at an LPS-induced MyD88 pathway in the intestines, and I'll go over a brief description here. So first, you have LPS that binds on the on the outside of the cell to a toll-like receptor 4. Then MyD88 binds to the toll-like receptor 4 inside the cell that activates the signaling cascade. This signaling cascade eventually allows for the transcription and translation of MLCK, myosin light chain kinase, that regulates cytoskeleton elements. So MLCK allows for the cytoskeleton to contract, leading to tight junctions opening and thus an increase in intestinal permeability. And to see whether this LPS-induced pathway is being affected, we are examining protein expression using Western blots for MyD88 and MLCK. I currently only have data for MyD88 within the small and large intestine. So here we have MyD88 protein expression within the ileum. And here I took the density of each of these bands between the two different groups, and there was no significant difference in the protein expression of MyD88, as well as in the cecum, there was no protein expression difference. In the proximal colon, there was also no significant difference detected, as well as in the distal colon, there was no significant difference in MyD88 protein expression. Now moving away from that, as I mentioned, the second portion was goblet cells. So we wanted to see if goblet cell number was changing based off HDE exposure. So I did a pass stain on different sections of the large intestine. Here we have the cecum. So the goblet cells are these circular magenta blobs that I would count. And then the purple dots are the nuclei within each crypt that I would count. And I would standardize that by taking the goblet to nuclei ratio. And here we have that between the two different groups, and there was no significant difference in the goblet cell number within the cecum. As well as in the proximal colon, there was no significant difference of this goblet cell to nuclei ratio, as well as in the distal colon. So just to summarize everything, so first off, 3-week HDE exposure showed that there was chronic airway inflammation in the lungs of these mice. There was an increase in intestinal permeability within the gut, and also the exposure induced endotoxemia. With my Western blot analysis and pass stain, There was no difference in mighty protein expression throughout the large intestine and small intestine. Also, there was no effect on goblet cell number within the large intestine. So some future experiments that I still need to do, I still have to look at MLCK within the pathway, but we are also interested in looking at CD14, toll like receptor 4, and MD2, because they take part in the process of LPS binding. Overall, this project, I hope to broaden the knowledge of how organs have effect on one another in terms of disease treatment and prevention. Thank you. Oh, also, I would like to acknowledge the Dr. McColl Lab, my PI, Dr. Declan McColl, and my postdoctoral mentor, Dr. Melissa Crawford, and also the Marquis Star for funding me with an NIH grant. Any questions?
Thanks, Brianna.